Hello and welcome everyone to this video podcast for human genetics. This will be our third podcast on inheritance. I'm going to begin by comparing monofactorial traits with multifactorial traits. And then I want to introduce pedigrees, a tool that geneticists use and we will use periodically throughout the semester. Okay, so let's go ahead and begin with monofactorial traits. These are also called single gene traits. Now, up to now, we've talked about Mendel's pea plants and why he chose those. And we talked about things like seed shape, seed color, plant height, and some other factors. Now, all of these factors that he followed, that he used to make his first and second laws, are monofactorial. One gene was responsible for the seed shape, the seed color, and the plant height. It turns out that traits that are caused by one gene, monofactorial ones, are rare. However, they are easier to study. Now, most traits are what we might call multifactorial. And later on in this semester, we're going to talk quite a bit about multifactorial traits and how we study them and the different factors that are important for multifactorial traits. But it is safe to say that most traits we see in humans are multifactorial. We can think of simple traits like eye color. There are many genes involved in determining someone's eye color. Our height and weight. We know that there are many genes that play a factor in height and weight. We also know that there are environmental factors that also need to be considered. We also know health conditions and diseases can be multifactorial. And I could list several, but let me just throw down something like asthma. We know there is a genetic link to asthma, but we also know that there are certain environmental factors that can make asthma better or worse. So it would be considered multifactorial. I should have said this when I first introduced multifactorial, but I want you to know that multifactorial traits are caused by a combination of multiple genes plus usually environmental conditions. And as I mentioned, we'll spend more time talking about multifactorial traits and how we study them. But even with multifactorial traits that are caused by multiple genes, often an investigator may only be studying a single gene involved in that trait or that disease. And so that's what I want to spend the rest of this podcast talking about. Because ultimately, if you're just studying a single gene, it will be easier to study, even if there are other factors involved. So what I want to do right now is introduce pedigrees. Okay, let's go ahead and begin our discussion of pedigrees. I'm showing you this picture of a pedigree just to introduce you to what a complex pedigree may look like. Now, right now, as you're looking at it, you may not know what the different symbols mean. You may not know what a circle, a square, or a diamond mean, what a cross between the different symbols means, if they're shaded in or if they're not shaded in, what the numbers mean, what these numbers on the side mean, what this arrow means. So what I'd like to do first is introduce to you what these different shapes and symbols mean. There's a table that explains these shapes in your textbook as well. I thought this table here had a few more components on it that I think are important. Let's go ahead and begin on the right side. Now, each individual in a pedigree will be identified as either a square, a circle, or a diamond. If they're a square, then they're a male. If it's a circle, then that individual is female. And if it's a diamond, that means that the sex was not known. Now, sometimes this happens if you're looking at an older pedigree and you know children were born, but you don't know if they were male or female. Or sometimes there are babies lost in spontaneous abortions and the sex wasn't determined at the time of the spontaneous abortion. Now, the difference between it being colored in or not colored in is an indication of whether or not that individual had the trait. If they are colored in, as shown here, that means they are an affected individual. They have the trait. And any given pedigree that we'll be looking at will only be looking at one trait throughout the pedigree. There won't be multiple traits within a pedigree. Now, sometimes in a pedigree, somebody may have 5 kids or 8 kids, and if they're all unaffected or all affected, sometimes they might just indicate it with a number. It makes a larger pedigree simpler to look at. If you see a line through here, that will indicate that the individual is deceased. I don't think we'll be seeing this in any of our pedigrees, but if you do see a P in a shape, that signifies the unborn baby of a pregnant mother. Typically, that means that the mother is pregnant, and if they know the sex of the baby, they will indicate it with a square or a circle in the pedigree, or they might just use a diamond if the sex of the baby is not known. A miscarriage is shown with a diamond, and then typically a pedigree will have one arrow on it like this. And all this arrow indicates is that this was the individual who provided the information for the pedigree, or where the pedigree investigation began. It's not shown here, but that is called the proband. Another thing that's not shown here is if an individual is a heterozygote. So let's include that here. If the person is known to be a heterozygote, half of the individual will be shaded in. Sometimes it's shaded in at the diagonal on some pedigrees. Sometimes it's shaded in at the horizontal, like so. Either would be acceptable. An important thing to remember when you're looking at a pedigree is that you only indicate if someone is a heterozygote if you know for sure they are a heterozygote. So for instance, you may look at a pedigree and one of these individuals may be a heterozygote, but it's not indicated just simply because we don't know. Because often if you're looking at a recessive trait, an individual may be a heterozygote, but you don't know for sure. And we'll talk more about that when we look at some specific pedigrees. Now let's look over here on the left side. When you have two individuals with a line connecting them like so, those individuals are said to be married. You can indicate a divorce or separation with lines between them. We will probably not see this in any of the pedigrees we look at, but if you do, you know why. If they are blood relative, we say there is a consanguous relationship, and we show that by a marriage line, but show it with 2 lines. We will see this, and usually when we see it, it is because of first or sometimes second cousins being married, but anytime there is a close relationship, it would be shown here as a double line. Now, to show siblings, we draw a line straight down from the marriage line, and then a horizontal line connecting to each of their kids. In this case, they'd had 2 sons. I should also point out, it's said here, but I didn't stress it. This line between the 2 shapes doesn't just mean they're married. They could just be in partnership. Now, we may see these symbols here showing identical twins, monozygotic, or non-identical twins, dizygotic, or fraternal, you may refer them as. And that is when you draw this line down, instead of drawing a horizontal line like this, you draw diagonal lines like so for twins, and if they are identical, you connect them with that line. And there's no connecting line here if they are non-identical twins. Okay, let's go ahead and now talk about the kinds of traits we can determine by looking at a pedigree. A pedigree will help determine the mode of inheritance. And throughout this semester, we will talk about several modes of inheritance, and we'll talk about how we can look at those using a pedigree. I'm going to list them now, but we're only going to look at pedigrees for 2 of them today. So the first 2 are autosomal recessive and autosomal dominant. We will talk about each of these today. Remember, autosomal means it's a gene that is located on one of the autosome chromosomes. It's not on one of the sex chromosomes. So the next few we're going to talk about are on a sex chromosome. X-linked dominant, X-linked recessive, Y-linked, and the last one we'll look at later on are mitochondrial traits. But we're just going to talk about autosomal recessive and autosomal dominant for this chapter, and we'll come back to these 4 later. Okay, so let's look at two pedigrees. On the left is a pedigree of an autosomal recessive trait. On the right is a pedigree showing the inheritance of a autosomal dominant trait. And let's first write down a couple examples of such traits. Sickle cell anemia would be an example of an autosomal recessive trait. Another disease called Tay-Sachs is an autosomal recessive trait. Something that's not a disease but is a trait would be the ability to roll your tongue. Some of us can do that, some of us can't. Some examples of autosomal dominant traits include Huntington's disease. And another example would be Marfan syndrome, a syndrome that we believe Abraham Lincoln had. Let's now consider some characteristics of each of these. The first is true for both of them. There's not a sex bias. Autosomal traits, you would expect to see roughly equal numbers of males and females with the trait. Now, the way you can see that is if you look at this pedigree here, you see 1, 2, 3, 4, 5, 6, 7 males, and then we see 1, 2, 3, 4, 5 females. It's a slight difference, but they're close. They don't have to be the exact same. But if we saw a pedigree, and we will see this when we look at X-linked recessive and X-linked dominant and Y-linked, we'll see a majority of either males or females with the trait. We can also look here, and you can see these two parents here had 4 kids, 2 with the trait, and 1 was male and 1 was female. Over here you can see a similar trend. You see 1, 2, 3, 4 females with the trait, and 1, 2, 3 males with the trait. So there's not a huge sex bias in autosomal traits. 2, this is a very important part of an autosomal recessive trait. The trait can skip a generation. So over here we have 4 generations, 1, 2, 3, 4. And when we say skipping a generation, we don't mean everybody in generation 2 across the line doesn't have it. We mean that you will have some members of that generation who don't have the trait, like you see here with generation 2, individual 2 here. They don't have the trait, but they pass it on to their kids, so it skipped that generation. Same thing here. These 2 individuals don't have the trait, but they passed it on to their kids. So it skipped that generation here. Now, it doesn't have to skip a generation. It's not a requirement. For instance, you have these individuals here who have the trait, both of them have the trait, and all their children have it. Over here, we say it cannot skip a generation. You see this individual having it, and they have a child with it. Same thing here, and same thing with the first generation. However, individual 3 here doesn't have the trait. Since he doesn't have the trait, he can't pass it on. Now let's talk about why that's true. Remember, if you have an autosomal dominant trait, say like Huntington's disease, we're going to show that as big H, little h. This means you have the trait because you have the dominant allele that leads to the disease. Then that individual will pass on either big H or little h to their gametes. When this gamete combines with another gamete, and let's just assume it's a little h, somebody who doesn't have the trait, that progeny will be big H, little h, and this progeny will be little h, little h. So 50% of the progeny here will have Huntington's disease and 50% won't. This individual now can go on and pass it on to the next generation. So let's say pass here, because I'm running out of room. This individual, since they don't have the big H allele, can never pass it on. So I'm going to put just can't. But in your notes, you might write can't pass it on. Once that disease allele or trait allele is now no longer in an individual, it's gone forever. It cannot be passed on. This individual here just got lucky, right? They don't have the trait, and so now they can't pass it on to their progeny. These individuals can. Over here, let's talk similarly. Remember, and let's think about Tay-Sachs. In this case, it's a recessive trait, so the big T indicates the normal, the healthy allele, and the little t represents the disease allele. So this individual is healthy. And if they have children with somebody else who is a heterozygote, a healthy individual, then you can do a Punnett square if you like, but we know that 3/4 of those individuals will be healthy. They will have that big T allele and either another big T or a little t. But 1/4 of them will be little t, or have the trait. And that's what we see up here. We see these two individuals. It skipped that generation, right? But they're both heterozygote. And how do we know they're both heterozygote? Because they have children that are little t, little t. In fact, let's just go ahead and show the Punnett square. So these two individuals here are the result of that. These two individuals here, 6 and 8, are either big T, or big T, little t. We don't know which one they are, but we know they don't have the trait. So when we look at this, what can we conclude about these two parents here? They both have the trait, so we know that they have to be little t, each of them do, because they have the trait, and it's a recessive trait. If they both have the trait, then all of their children will also have the trait. And if you need to, you can do a little Punnett square here to convince yourself of that. You can see when you do that, all of the progeny are little t, little t. It would be impossible for them to have a kid that doesn't have the trait. Okay, I want to talk about a couple other characteristics. And some of them we've already talked about, but I want to put it in writing. So 2 affected parents will always have affected kids, as is shown here. Over here, what we'll say is that an affected parent will pass the trait to half their kids. Similarly, as we showed before I erased it, the Huntington's disease example. And now this is only true if the affected parent is a heterozygote, like that. If they are homozygous dominant, then all of their kids would have that trait. Now, in practice, homozygous dominant individuals for autosomal dominant traits are extremely rare. Usually, these kinds of traits are lethal, particularly in something like Huntington's disease. For our purposes in this class, if I'm talking about an autosomal dominant trait, you can assume that they are heterozygous, unless I state otherwise. But if I don't state otherwise, assume it's heterozygous. Now remember, I mentioned that heterozygotes, if they are present in a pedigree, they're not always shown. So for instance here, we know that both these parents have to be heterozygotes because they had an affected child. So don't be confused when you're looking at it and you don't see something half shaded in, because it doesn't always show it. That leads me to my last characteristic here of autosomal recessive traits, and that is parents of an affected child must either be affected themselves, okay, so we can go like little a, or heterozygous. So it might be like big A, little a. If one of the parents is big A in this example here, then they would not be able to have a child that is affected. So both parents have to either be homozygous recessive or heterozygous. So for instance here, these two parents here, we know that person's shaded in, so we know that they must be little a, or homozygous recessive. And since they had a child that had the trait, we know that number 6 here has to be heterozygous. And if you're not sure why that's the case, let's go ahead and do a Punnett square. We know 7 here has to be little a. If 6 were big A, these are the progeny they would make, all of them unaffected. So since we know this child's affected, we know that number 6 has to be heterozygote. So they would be little a, they could have children that are little a. And that's what you see here. As practice for you, to make sure you understand everything in these pedigrees, go ahead and try and indicate all of the possible genotypes for these individuals. You should be able to make estimates on what their genotypes are. And if you do that, say take a picture of this and write down the genotypes that you're predicting, you can email that to me and I'll tell you if you're correct. And I would do that with the autosomal dominant trait as well. Okay, well that ends this podcast, and it's our last podcast on Mendelian inheritance. In this podcast, we talked about the differences between a monofactorial and a multifactorial trait. And we introduced pedigrees, described what all the shapes and symbols mean, and we also talked about how you could use those to make a prediction if it's autosomal dominant or autosomal recessive. On an exam or an activity during the week, it's very likely you will see a pedigree and you'll have to tell me, is it autosomal recessive or is it autosomal dominant? So those characteristics that we went over, think about those characteristics and make a prediction about whether it is autosomal recessive or autosomal dominant. All right, let me know if you have any questions. If not, I'll see you on the next podcast. Bye now.